Good morning and welcome to day two of the Future of Biopharmaceutical Analysis Summit 2026. Start off today with a really fascinating panel on lightening the nitrosamine load, sponsored by USP. Nitrosamines have long been a bit of a bugbear of the industry, and our panelists today have a huge range of experience. You have the opportunity to ask questions all the way through, so if you pop them into the chat on the Swapcard platform, we will then get them to the end. Our aim is to leave a few minutes at the end for questions, so keep them coming in throughout. Our panelists today are Andrew Teasdale, Director and Owner of ATC MC Solutions Limited, Archana Bahugunne, Regulatory Compliance Specialist, Malcolm Rossi, Analytical Specialist, External and member of the Nitrosamine Task Force for Novartis, and Mayak Banti, the Senior Director at USP. And I just want to start with a bit of an icebreaker. So Malcolm described himself recently to me as possibly the only person in the industry who finds nitrosamines genuinely wonderful, largely because they've kept him employed almost indefinitely. So before we get into the substance of the discussion, and I'll start with you, Malcolm, I think it's only fair that you get right to reply. I'd just like to ask you to answer in a sentence or two. When you first encountered the nitrosamine problem, did you realize what you were getting yourself into?
No way. I thought this was a one-off Valsartan, stupid company mucking around with processes out of control, and a stupid regulatory authority who had decided that they were going to make an issue out of it. Boy, was I wrong.
Shana, what about yourself?
Definitely. 8 years back, when we first heard about the started cases, it was like a lot of curiosity, a lot of questions, very few answers. And little did I know that 8 years down the line, uh, a small impurity would change or would transform the entire regulatory landscape globally. So it has been an enriching experience over the years to have been part of the initiative that drives this change and to look at how global engagements and collaborations can transform lives of patients.
Brilliant. Mayank, what about yourself?
For me, if you really see when this whole issue of nitrosamine unfolded, it was around 2018, 2019, and I joined USB in 2019. So I still remember sitting in those early discussion, trying to grasp what was happening from a patient safety perspective. At that point, honestly, it felt like a contained issues. We didn't imagine it would go beyond much ARBs. So, but I think very quickly it has evolved, and that's where I think USP has also promptly constituted that expert subcommittee who has helped us to develop this general chapter. But to answer your question, yeah, initially we thought that it is just a contained issue related to ARBs.
Andrew, how about yourself?
Yeah, I mean, I think some of the key points have already been made by others about what we thought in the first instance about what the likely scope was. I suppose my first reaction to it was, well, how on earth did they manage to end up with a nitrosamine in the process? Because from a chemistry point of view, nitrosamines are certainly not something you would ever use as part of a controlled chemistry. So the first question was, how on earth were they there? Well, that then got answered. I think, also as well, it would very quickly became apparent that this issue was snowballing. It was valsartan, then within a couple of weeks it was irbesartan, and then there was a contamination issue with pioglitazone, and that led to this almost inevitable snowballing as regards this issue. And the interesting thing was that when we started to have to do the investigation triggered by the regulatory authorities on every small molecule, in the first instance, and the irony is, in the first instance it was focused on drug substance chemistry, we would have never actually discovered what ultimately has turned out to be the real elephant in the room, which is not the small nitrosamine seen in the chemistry. It's the NDSRIs formed in the drug product. And that was the interesting thing, how quickly it evolved from one issue that was chemistry related, which actually we fairly quickly understood and were able to control, to a much, much larger issue of formation of nitrosamines within the drug product. And that is really where we find ourselves now with that second more significant challenge.
If I can add a personal note.
Go for it.
I'd like to add a personal note. If you look at my desk as it was in Novartis back in 18, 19, and 20, you would have seen sheets of paper covered with zeros and crossing out. I never ever had to deal with ppm, let alone ppb. And whenever I did the calculations, I was absolutely petrified that I was missing or moving a decimal place the wrong way. And I literally used to sit there with these sheets of paper going, no, cross that out, cross that out, cross that out. We just weren't used, I think, in the analytical terms. The pesticide people were used to talking about ppbs, but I don't think in the pharmaceutical industry, I mean, 0.1%. Well, that's huge when we started to talking about nitrosamine. So mentally, this was a challenge as well.
Mm.
Yeah,
And
it
I
took
think
it
that's an interesting thing to kind of touch on. And I just want to explore that kind of transition from where we were in sort of 2018, 2019 to where we are now, and how it's kind of changed a little bit. So when it first kind of crossed your desks, it was basically firefighting, and I'm kind of hoping that since then we've moved on from firefighting to a more systematic approach. So Andy, from your kind of vantage point, and you've been in this pretty much the whole time, what's the single most significant shift in how the industry is approaching this over maybe the last 12 to 18 months?
Well, I think the thing in the first instance was very quickly industry realized that we had to work collaboratively and actually better understand the science associated with nitrosamines. And that understanding, particularly in the context of the chemistry, now means that we can largely avoid the formation of simple dialkyl nitrosamines in the drug substance by controlling the relevant factors. And that actually the original issue is not such a significant risk. And I think it is that science and understanding that has allowed us to be systematic in the way we do the risk assessments. And we've continued with that and developed the workflows through FPA for drug substance and drug product. And these workflows work very well in terms of being able to address this systematically. The trouble comes, even though now we have a defined process for defining limits, the CPCA, if you follow this process, you do the CPC calculation and you find that your NDSRI in your drug product is category 1 or category 2, then we have a really significant issue. And that is still the case. And actually, it's got more complex by the fact that we are continuing to identify, unfortunately, other risk factors. And this is coming from the science. I mean, we know, and we'll almost certainly come back to the topic of NOx in this discussion, because that's another vector in terms of where nitrosating agent may come from. So yes, we developed a systematic way of doing it, but it's not clear even with the systematic approach that we get to the end and actually find ourselves in a manageable position, particularly for NDSRIs.
And I think the interesting thing is with that kind of evolution and with that kind of level of progress and systematization, it also puts kind of pressure on the regulatory requirements. And Archana, I just want to turn to you for this one. So, the regulatory requirements around nitrosamines have continued to evolve, as they should. The FDA in particular has been updating expectations. But kind of broadly, how is the global regulatory scenario with regard to nitrosamines? And are the kind of the deadlines and requirements becoming clearer, or is there still significant uncertainty that manufacturers have to navigate?
Thanks, and thanks, Ian, for having this at the start of the session. So, as we discussed earlier, the global regulatory scenario has been evolving over the past 8 years. Initially, when we started with the sartans, post sartans, it has been a situation where we have been building the foundation blocks, building the foundation, answering questions. Uh, where we stand today is in a transitional stage where we're moving from building the foundations to a more kind of a structured regulatory framework for assessments, testing, and mitigation of nitrosamines. I don't say we have completely transitioned, but if we see the history, it started with basically, the guidelines from the regulatory agencies. Subsequently, we saw once the guidelines were established, when there was some data available at hand, the pharmacopoeia started getting into action with respect to publication of compendial drafts. So, if we see the situation today, we've seen in the past frequently the scope of nitrosamines and NDSRIs gets updated in the guidelines issued by various agencies across the globe. The AI limits get updated from time to time. Um, Talking about the pharmacopoeias now, initially what started with a general requirement with respect to control and testing of nitrosamine impurities. We've seen in the past that the USP general chapter is under revision and was open for comments till January this year. We've seen the Pharma Europa 12th edition is now effective. About 2 months back, it got effective. And the scope of the general chapter of nitrosamines was earlier restricted to active substances, but now it extends to drug products. As we discussed this session today, 7 days down the line, the 10th edition of Indian Pharmacopoeia gets official and effective, and is the first time that the general notices and general monographs and the general chapters of Indian Pharmacopoeia make it mandatory for testing of nitrosamines and LDSRIs. Now, does that impact only the Indian context? The answer is no. It's just not India, but also the pharmacopoeia has a recognition in about 24 nations across the globe, spanning geographies. So even those agencies and the patients in those countries get access to medications which are controlled with respect to nitrosamines. So if we see this scenario, we would say that the global regulatory scenario has been evolving. Talking about the current year, earlier in this year, USP launched a new initiative with respect to MICF, that is Mutagenic Impurities Compendial Framework. We had a workshop, first a round of workshop a couple of months back in this aspect. There's also a lot of focus, I would say, with respect to excipients. As we go down during the discussion in the session, we will talk about how excipients and its impurities can have an impact with respect to nitrosamines and NDSRI. So we see information chapters being published by the Pharmacopoeia with respect to CQAs and impurity profiling of excipients. Um, Last year, we saw WHO updating its Annex 3 with respect to WHO GMPs for pharmaceutical excipients. And it's interesting to note that the Annex 2 in this particular guideline talks of high risk excipients, and nitrosamines are listed in over there. So the regulatory scenario is evolving. Uh, we are more moving towards a structured framework. Talking about the timelines, that August 2025 timelines that were earlier prescribed by FDA, they are now kind of valid with respect to submission of risk evaluation and confirmatory testing. However, with respect to risk mitigation strategies and implementation of CAMA plan, the agency did take a pragmatic and flexible approach with respect to extending the timelines. With respect to implementation of CAPA plans, and manufacturers are now required to submit progress reports with respect to the proposed plans and the timelines. So, what I would also like to add is that there has been a lot of collaboration and engagement between the industry, the agencies, and the compendial authorities, and that is how the whole matter has moved from a primitive stage to a far more advanced stage today, where we have relatively better controls over the whole issue. We are still not done with it, but definitely we have better issues, and there's a long way to go ahead with it.
Mayank, I think it's an interesting one for USP because you're in a kind of interesting position here. You see the demand for reference standards and analytical methods across pretty much the whole industry. Has that demand picture kind of stabilized over the last year or so? Or are manufacturers still coming to you with fundamentally new nitrosamine problems that you haven't seen before?
I would say that the demand has evolved rather than fully stabilized. We are no longer just seeing the known nitrosamine issues, right? I mean, manufacturers are still coming up with new, more complex challenges, especially around unexpected formation pathways and novel nitrosamine structures. So while there is a maturity and preparedness compared to a year ago or a few years ago. The problem itself has not plateaued. Yeah. It's shifting and in many ways becoming more and more sophisticated, I would say, and keeping us all on our toes. In terms of demand, I think the number of NDSRIs, pharmaceutical analytical impurities in our catalog has increased to more than 150. And we are continuously getting more requirements from the manufacturers. But apart from the impurities, USP's nitrosamine analytical framework is designed so that we can decode this whole nitrosamine puzzle. And it consists of both the compendial solution as well as the non-compendial solution. So on one hand, the compendial solutions provides the documentary standards and reference standards. The non-compendial solutions provides a solution to more evolving challenges. For example, we have the issue of determining nitrosamine in solvents, or mitigating some of the matrix interference issues in LCMS, or maybe the issue related to NDSRI characterizations, and most recently the nitrite determination in excipient. So we publish these in the international journals as well as in our analytical hub, which is hosted in nitrosamine exchange community. So the demand is evolving, as I mentioned.
It sounds like it's all getting more complex, and I think that leads nicely to the broad question of whether the challenge is really diminishing or growing. I suspect that I know the answer to this one before we start talking, but Andrew, Meg touched on NDSRIs, but the original nitrosamine problem, as I understand it, was around NDMA contamination from manufacturing processes, which, when you think of it as a problem, it's a solvable problem, it's a tractable problem. You find the source, you eliminate it, and job done. NDSRIs are a bit of a different beast because the impurity is potentially being generated by the drug substance itself. And again, do correct me if I'm wrong, because I am not a nitrosamine expert.
No, that's correct. Yeah.
Yeah, good.
Sorry,
sorry, carry on.
No, I was just going to say, I mean, in terms of the issue itself, so you're right. I mean, the issues and challenges around the simple dialkyl nitrosamines, through our understanding of the mechanism involved, we can actually accurately model whether or not you'll form a nitrosamine in the drug substance. And there's other really important factors in regards to this as well. And if you look at Mayank's background, he has a triangle. And with nitrosamines, we now often talk about the nitrosamine fire triangle. You need secondary amine. Tertiary amines are far less of a risk unless they're activated. You need a secondary amine, you need a nitrosating agent, and you need the conditions. Now, we can avoid, in the context of the drug substance, many of those things. And another important factor is we can analyze for most of these simple dialkyl nitrosamines. USP has provided a suite of methods that allow us to do that. And, we can almost take a generic approach to risk assess them, also to test for them. And we know what the limits are for those. But the NDSRI situation is far, far more complex. We know, for instance, whilst it's unlikely we use a nitrosating agent typically in chemistry, if your drug is a secondary amine, you could get exposure in your API manufacture to NOx. You could form a nitrosamine there. Operations like air-jet milling introduce air as well, and potentially these NOx species become relevant in the context of that. Also, as well, when you move to the formulated product, then of course you've got not only the formulation, the operations like spray drying and other drying processes. We also, of course, have the fact that we know we introduce nitrites as a consequence of the excipients we use. Yes, we've driven those down, but actually, I'm sure when we get into some more of the discussion, we actually know there's some nuances as regards that. Are we truly measuring the right level? And actually, even if you have a low nitrite level in your excipient to start with, is that always true when you move through into operations like fluid bed drying? And so there's a lot of complexity around the NDSRIs that we simply don't have with the nitrosamines in the API. They're avoidable. So we've made massive strides with that. The NDSRIs, we're still learning, I'm afraid. And the final thing I'd say on that is, in the case of the simple dialkyl nitrosamines, we know what the limits are. For the NDSRIs, for a long period of time, we didn't even have any idea what was an acceptable limit. We have now the CPCA, the carcinogenicity potency characterization approach. That's made a difference. It's stopped us losing lots of critical drugs. But let's not pretend that is a perfect solution to defining limits, because it isn't. And the preclinical science dramatically needs to improve as well and get more consistency around that. So yeah, for me, it's more complex in the area that is the bigger risk factor, which is the NDSRIs. And I think, that probably defines where I am with this at the moment.
So if it absolutely has become more complex, then I think that's really clear. I just want to delve a little bit into the analytical side on that. And maybe, Mayank, this is one for you. Are our testing methods now generally fit for purpose across the range of nitrosamines we're dealing with, including NDSRIs, where certified reference standards may not exist in every case, and where are the real remaining gaps in our ability to detect and especially quantify accurately?
I would say that we are in a much better place analytically than we were a few years ago. Methods today are far more sensitive and broadly applicable. For many classical nitrosamine, we do have fit for purpose method supported by reference standards, and that's brought a lot of confidence. Where the gaps still remain around the robust method validations. Typically, uh, samples are prepared by total dissolution or precipitation or extraction, etc. But the recovery studies are performed by liquid spiking, means that API or a sample is weighed in a volumetric flask, diluted with a diluent, and then spike level solution is used for making up the solution. Now, though there would be an interaction between the API and the nitrosamine impurities in the spike solution, but probably this would not represent the true recovery. So we need a solution for it which can help us to understand what is the true recovery is. One of the solution for this is maybe we can have an artificially contaminated sample, which can be prepared, by first spiking the known level into a powder sample probably, and then make a slurry with a suitable solvent, evaporate it, and get a free flowing sample. So that will kind of imitate a real sample with known level of nitrosamine impurities. Now, this sample can be used for checking the recoveries in our methods, and that will give us a true reflection of the recoveries and whether our method is capable enough to detect at that less or ultra low level. And by doing this, we can avoid the problems which we may face when we are analyzing the real samples. Apart from that, as Andrew mentioned, NDSRI is completely a different complex zone. And what is the major issue there is that NDSRIs are very unique to API. And if we have to develop a method, first we have to synthesize these impurities. And for that, typically, different formation pathways needs to be used, maybe a nitration reaction. Now, during this reaction, there could be a chance that a C nitration may happen or an O nitration happen along with N nitration. So characterization plays a very important role. So it is very difficult to say that by a single technique uh, we can confirm the formation of NDSI impurities. Maybe orthogonal techniques, including the NMR, the 2D NMR, can be used for that purpose. And then we can have some confirmation of these impurities, and then it can be used for the method development. In fact, I think USP has recently published one research paper on characterization of NDSRIs and has proposed a workflow for characterizations that may really help to do a correct uh, a confirmation of these impurities. So while we have improved on the method, but still a lot need to be done in terms of validating it properly and then using it for the analysis.
If I could just very, very quickly add to that, one of the other challenges with NDSRIs is sometimes the sample preparation is really critical. You could develop an analytical method for one form of the product. A metformin products are a classic example. You know, if they are immediate release, the analytical method is relatively straightforward. If they're sustained release, it is much, much more complicated. We've seen false positives, false negatives, and so unfortunately for NDSRIs, you know, it clearly is a case of one size does not fit all. It's not even a case that once you develop a method for the specific NDSRI, that it'll work for all matrices. You know, you've got that dependency on the matrix, which is just making this so much more complex. And so, you know, the holy grail would be, can we find a more systematic way of doing the sample preparation as well within this? I think that's an area that I'm particularly interested in, but I'm not sure we're not there at the moment. But Mayank or others, I don't know if you have any reflections on that, but that's certainly, I know, another area of big concern for me personally.
Yeah.
Can
So
I? Okay,
Yeah,
so
yeah.
go ahead.
now, what I was just trying to add here is that as we know that NDS are very unique to API and the structure is very similar to API. So sometimes it's become challenging to have a chromatographic separation as well, the required resolution, and that also makes the life difficult. And in fact, since we have to achieve low levels, so we use mass spectrometry methods platform. So even there, if you're using a high resolution mass spectrometer, we see challenges when we try to establish the quantitative ions, qualitative ions. There could be a possibility of isotopes within NDSRIs, and that creates another complexity. So we get a lot of questions around it, and you know, it's very important to understand how the spectra can be interpreted effectively.
Welcome.
I'm less optimistic than my colleagues. A lot of my activities are related to investigation of root cause, and we frequently find ourselves hampered by the lack of sensitivity. Um, you tend to be talking about analyzing the finished product as it is on the shelves, setting a specification, which is precisely the safety issue. But we are looking at what's the kinetics of the development in order to identify what the root cause is. This means we're looking at levels which may be considerably lower than the acceptable limit, but we need to see this in advance. And of course, unfortunately, with drug products, the NDSRI does not appear immediately. I wish it would, but it appears over a period of time. So we're also running into stability issues where at time zero, we have zero or below LOD. 2 months later, we've suddenly got an NDSRI has appeared. Now, the almost every single case that I've been involved with recently, we are sitting and looking at results and we are saying, now, is this analytical error or is this a real, genuine result? This particularly applies to the NOx, which we've been doing a lot of work with recently in fluid bed dryers. It doesn't happen when you start. You put your granulate, your wet granulate into a fluid bed dryer, you dry it, you analyze it, and you say, oh, look, we have no NDSRI. Go away, have a cup of coffee, and you'll then come back and you'll find your NDSRI. The other problem is because of the analytical error, which when we're down to the PPBs, the analytical error at the moment with the analytical methods is pretty large. It's very difficult to construct a genuine kinetics in order to predict not what's happened over the 2 months or 3 months. This is the old problem of stability. Can you really rely on the Arrhenius equation, which we all know is very, as my first professor said, it's useful for setting exam questions to students, but not much more. We really don't know. we're looking at situations where we have to maybe monitor for 6 months or a year before we can get to a level of NDSRI where it isn't masked by the analytical error when we're down to the LOQ and LOD. And I would say that is a huge analytical challenge. I totally agree about sample preparation. If you're doing formulation studies, every single matrix requires a revalidation because of the extraction methods. And please, just a general word, will people not use ultrasonic baths? Because we have proved conclusively. We have some interesting figures, but you still see papers being published using ultrasonic baths. Ultrasonic baths generate nitrite and in an uncontrollable way because it depends if the solvent is water or if the solvent is methanol, and it's not necessarily in the direction you predict. So yes, absolutely. Sample preparation is a huge problem, but even with the solution to sample preparation at the low levels that we're using for root cause analysis, we want to see what happens when you do this, what happens when you do that. We are still in the sort of area of analytical error for some considerable time, and we would love an improvement in analytical sensitivity.
I'm
I think
glad,
that
Malcolm, you have raised this point of analytical error, and that goes back to what I mentioned earlier, that uh, the methods needs to be validated effectively, and we need to have a risk assessment. The kind of attention placed.
Sorry?
I'm sorry, there was a fire alarm testing going on, so
I hope it was only a test.
yes.
yes,
Please do feel free to run out of the building if you have to, Mayank.
no, I think this is fine.
Excellent. So I think that leads nicely on, and you just touched on this, Mayank, to kind of compliance and compliance failures and struggles. So this is obviously an area where there are lots and lots of pitfalls and lots of struggles around compliance. Shana, maybe you can answer this one first. So what are the most common failure modes that you're seeing? Is it primarily technical testing, risk assessment, something more organizational? And how can these challenges be addressed by firms?
I think that's a very interesting question, and I think some of the answers we've already had earlier during the session. We're talking about NDSRIs, we're talking about their mechanism of formation, analytical issues. Now, what's interesting to understand is that we're talking of developmental issues when a product and a process and a method transfers into a manufacturing facility which is working under GMP environment. We cannot tell agency that I don't know how it is happening. I have I have NDSRIs, I have methods which are not performing well, and I don't know how to address it. So, uh, what I'm trying to communicate is that we are in a stage where I would say development versus compliance, the development, understanding of the process, understanding of the risk factors, understanding of the methods, understanding of the excipients, raw materials, their CQAs, how they impact the product. These experiments and these understandings are still underway. So if you were to ask me what exactly would lead to a compliance failure, I would say it would be an amalgamation of all these factors. So, if you see from the CGMP perspective, there are well established guidelines and protocols for manufacturers to comply to. So, the kind of failures it could lead to when we talk of process, the first and foremost would be inadequate risk assessment, no assignable root cause. Say, for example, no assignable root cause for a failure with respect to formation of particular NDSRA. Suppose you have a OOS at say about 18 months in a stability program. What do? You go back and start re-engineering the whole thing, trying to understand. And what does the investigator give you? That there is no assignable root cause for this kind of a formation. Similarly, when you're talking about nitrites in excipients, you have process being executed in your manufacturing facilities, and suddenly you find out the root cause is due to a nitrite in so and so excipient, the agency would come to a conclusion that you do not have adequate vendor qualification procedures in place. So that is how it gets classified, and compliance is an issue because we need to understand that nitrosamines, when we talk about nitrosamines, these are issues which are of public health concern, and that's why regulators are very stringent about it. So from the compliance perspective, I would say first one would be inadequate risk assessment leading to no assignable cost for root cause analysis. Say, for example, nitrite containing excipients, use of recycled solvents. That's the biggest sin that a manufacturer could do with respect to this topic. Inadequate specificity or sensitivity of analytical methods being employed by the manufacturers. There could be issues with respect to how you are doing your packaging screening studies. Supposing we've seen examples of migrations of nitrite from the printing inks into the primary packaging, and then contaminating the entire product. So issues with respect to inadequate investigations on this aspect. So these are concerns, I think, from the manufacturing perspective, and the biggest concern would be how adequate are your contamination control strategies, because when we move from lab scale to manufacturing scale, there is a concept of cleaning validation, cleaning processes. So you are having batches being manufactured one after the other. How adequate are your cleaning procedures to ensure that there are no traces of any precursor amine or nitrosating agent from the previous batches reaching into your next batch? And we saw in the past, we've seen a warning letter, FDA 483 being issued to a manufacturer of a certain medication to this effect, inadequacy of cleaning procedures. So you can imagine we already have a certain issue, and then we adding more fuel to the fire by having inadequate procedures. So I think these all things are important. Coming to the organizational part, this was more from the executional part. I think organizational part is also equally important. So when we talk about an organizational part from the GMP perspective, the questions would be with respect to what kind of trainings have you imparted to your personnel who are involved in risk assessment, testing of these impurities. So I think these kind of aspects would also be very important, because when we talk of confirmatory testing, we need to understand it's a very critical parameter in the entire three-step process, because that decides the further fate of decision. And if this testing itself is inadequate, many times we see the root cause. The analyst error, the analyst was trained on this. It doesn't work that way. We cannot accept or reject batches on the basis of such kind of justifications. So I think these aspects are very important to be understood from the compliance perspective, and who knows, going forward we may see a guideline or a kind of mandate from agencies with respect to what kind of controls they need to have with respect to this kind of contamination.
Andrew, I think Arshana touched on cleaning there, and I saw you nodding. Did you have anything specific that you've seen that you'd like to add to that point?
Yeah, well, I mean, I'm working with a client at the moment who've asked me to look at the whole aspect of cleaning in the context of nitrosamines, and I've spent a lot of time very recently looking through all of the various guidelines from the major regulatory authorities, FDA, EMA, WHO, as well as industry position papers from APIC and things like that. And it's really interesting when you look at them in terms of what are the expectations. And the expectations are not totally clear. In the main, the suggestion is, and Arantxa mentioned this, is that you're looking at this in the context that you could carry over precursors, a secondary amine or a nitrosating agent into the next product. But actually, a more challenging aspect is one that has come through this query from a regulatory authority, which is prove that you're not carrying over a nitrosamine. Now, for me, that's virtually impossible.
Yeah.
If you think about it, if you had a nitrosamine that was controlled in a secondary amine product as the guiding substance, you would clean that down to parts per million. The nitrosamine within it is already controlled to parts per billion. How could you possibly analytically prove that? Because, Malcolm's already talked about the analytical challenges and where we can go at the moment. We can go parts per billion, but we're not going parts per trillion or whatever is the level below that, picograms or femtograms or whatever. I mean, we're into the realms of homeopathy when we start getting to those levels. And so, for me, the real concern is that in some cases, the guidance is not particularly clear, and therefore it's open to over-interpretation and requests for approaches that actually aren't practical. And so, this is something I'm having to work with a client to challenge this and say, well, that's not what the guidance says, and these are the reasons why you can't do it. But I mean, well, the other deficiencies that Ranchia mentioned as well, to me, the best way to start is use a systematic approach. And I think the FPA workflows are really good at doing that, but they're not the total answer because there are other risk factors that we're still learning about, like fluid bed drying, NOx. We don't know the whole picture there. We know about reagents that actually contain NOx as well. I'm aware of the fact that the FDA are concerned over the level of NOx and nitrosation as a result of activated charcoal. To be honest, I'm not surprised by that in the slightest. Anybody that knows anything about charcoal knows that it's a bizarre material that can absorb just about everything. And so we can do all of these things, but there's still the interpretation that adds a layer of complexity, and there's still new risk factors that we still need to get to grips with. So, we're still on this conveyor belt towards, I don't know where we're on a conveyor belt towards, but we are on it. And, and maybe I'll just leave you one thought is that all of this comes about because we're trying to control the safety levels that are based on a very, very conservative approach. And I think ultimately the way forward on this may not totally come from a CMC control point of view and analytical measurements. A lot of it needs to perhaps come from a more rational look at what is the true safety risk, particularly for the NDSRIs.
Absolutely agree with you.
And I think that takes us nicely into the next topic I just wanted to cover off is we've looked at the history and we've looked at where we are now. So let's look a little bit about where we're going to as well. So Malcolm, I think this is one for you. So where do you see control strategies heading over the next 5 years? Is it more upstream formulation level controls, better by design approaches, or will just straightforward testing remain central for most products?
I'm going to distinguish between the current status of products on the market, commercial products, and that's going to stay exactly as it is. It's going to be endpoint testing. There will be more surprises about stability. I think there will also be some surprises about catalytic effect of excipients. I always remember when I did my PhD in chemistry, we used silica as a catalyst. Now, it's all very fine. We all ignore silica in the finished dosage forms because it's only 1%. But if I remember correctly, my definition of a catalyst was that it's not the amount that's there, it doesn't get used up. So I think we're going to find some excipient interactions which are going to come along. In the wearing my hat in the new products, the innovative products, I am not speaking specifically for any company here, but I believe we are going to get into a risk aversion scenario where people, because and Andy, you didn't mention, but it's not just the API, it's the impurities of the API, the degradation products of the API. So it's a whole plethora of other molecules, which when you're talking at the ppb levels, a 0.1% contaminant could become a critical issue. Now, I think we're going to see that development teams are going to be putting in more chemists. to evaluate the drug substance and the potential risk for nitrosamines. And I really, really horribly think that we're going to find that companies are going to adopt an over conservative approach, and we're going to end up with throwing the English expression, throwing the baby out with the bathwater. Um, at one stage, a couple of years back on one of these forums, somebody said, how can we avoid nitrosamines? And the answer is, well, don't have any nitrogen containing drug substances, which is true. I mean, that's one way of doing it. But then you throw away the whole pharmacopoeia. That's going to be, I think, a sad side effect where absolutely good drugs are going to be abandoned at the preclinical stage because of a potential, not a real, a potential nitrosamine risk executed by a chemist who doesn't necessarily understand that not all reactions that occur at 120 degrees under reflux in toluene necessarily are going to be reproduced in the drug product. So I think we're going to lose drugs. I think this is very sad, but I think we're going to lose potentially good drugs. There, I think the solution is back to something Andy mentioned, a more realistic interpretation from CPCA and determining real risk figures. I think the introduction of more logical approach to lifetime evaluations. We at the moment are looking at a product which is taken once in your life, but you have to do a nitrosamine assessment. So I think it's a combination of pressure from the regulators to make life a little bit easier for the industry, to leave the door open, because at the moment what I see is we are going to lose, unfortunately, nitrogen and amines are very common in drug molecules. And if we start excluding anything that's got a secondary amine or a potential secondary amine, when we go to choose our next clinical product, I think we're going to lose good drugs.
Just if I could very, very quickly comment on that as well, because we're talking about nitrosamines at the moment, but anybody that's seen the most recent version of the European Medicines Agency guideline will know that other nitroso species are also.
Oh, yes.
And, that is a specter that, still really, really concerns me. And particularly if we don't learn some of the lessons that, we've learned already with nitrosamines. So I do worry that even when we maybe make progress on this, we're just waiting for the next train to come into the station, and these other nitroso species, where in reality, frankly, we have even less knowledge of how they form, under what conditions, what's an appropriate safety limit, or all of these factors. So yeah, there's sadly a lot to be concerned about, I fear, and it could have the effect that Malcolm has said. We could just say, well, I'm just not going to develop these drugs, but then you're throwing away a huge rich theme of druggable molecules if we were to do that.
And Andy, I just had exactly one of these for a client, not Novartis, where the regulatory authority started to claim that there was the potential for a nitrosamide. Now, there's no nitrosamide anywhere listed in any of the lists of the EMA or whatever. It's an open subject. And what made it worse was it wasn't even an amide. But the company, which is one of the smaller companies, launched into a huge campaign of activity when they should have just turned around and said, get yourself a book on chemistry.
It's always a pleasure to talk to you guys, but there always reaches a point where I'm kind of fearful for the future, and I've just about reached that point. So I'm going to turn now to something which may be a positive for the future, which is looking at computational toxicology and AI and how this might impact. So May 8th, there's been a lot of interest in computational tools, AI driven, as everything is now. especially around predicting nitrosamine potency and setting some limits. How much confidence can we actually place in those tools at the moment? And what would need to be true for them to be kind of regulatorily accepted?
I would say that for the computational tool, there is definitely a growing confidence in these tools, but I feel that we are not at a point where they can be used in isolation. Right now, QSAR and AI-driven modes are very valuable for risk prioritization and bounding the problem, especially for NDSRIs where experimental data simply doesn't exist, right? So they help us make scientifically informed decisions rather than defaulting to overly conservative assumptions. But having said that, the confidence is still context dependent. Different models can give different predictions, and there are uncertainties around applicability domains, especially for more complex or the novel structure. So regulators and industry are still using them as a part of a weight of evidence approach and not as a single confirmatory answer. So I believe that for these tools to be more broadly accepted from a regulatory standpoint, a few things which are needed are like we need to have a greater transparency and standardization in how models are built and applied, cross validations and benchmarking across multiple models, more case studies where prediction aligns with the experimental or clinical data. So that will definitely instill more confidence. And importantly, clear guidance on how to use these outputs in setting limits. So these things are required. So in short, they are incredibly useful and increasingly influential, but we are still in that transition phase from supportive tool to fully regulatory anchored approach.
Ian, if I could just very quickly add to that, what's really interesting as well is some of the evolving preclinical data as well. And there's a particular paper that was produced by Bob Jolly of Eli Lilly recently, where he's compared the results of the revised Ames test, the enhanced Ames test, to the outcomes of some of the in vivo studies, the transgenic studies and other things like that. And what's really interesting is it now seems to validate the fact that if the enhanced Ames test is negative, that the in vivo study is negative. So one of the things that I would like to see would be, can we see, a more pragmatic approach being taken that actually goes back to some of the positions we have in ICHM7, where, for mutagens, before we started talking about nitrosamines, if we showed it was negative in the Ames test, then you tried it as a normal impurity. Now, I don't see that happening very quickly for nitrosamines, but that paper from Bob Jolly really seems to show that actually the Ames test is a very strong indicator of the potential carcinogenicity of a nitrosamine. So I hope we're going to start to be able to use that data in a more rational way than we are at the moment. So I think there is, on the safety side of things, there is some, hope. There is, as Mayank has said, some really nice in silico models now being developed, quantum mechanical calculations, looking at docking into enzyme cavity. It's really, really fascinating, and it does present a way forward. But I think we also need to sort some of the safety preclinical testing and the hierarchy of those as well to truly make progress. But I'm a bit more optimistic in those kind of areas than I am that we're going to totally control this purely and simply by using excipients with S-nitrate and doing everything under nitrogen without the possible chance of NOx being present. And yeah, I think the safety side of things is the one that really offers in some ways more hope.
Yep, I totally agree.
Charlotte, I just want to turn to you for this one. So looking at 2 or 3 years, what kind of categories of NDSRIs, and we've touched on this earlier, what types of products are not getting the attention they should? So where is kind of the next wave of compliance pressure likely to come from?
That's an interesting one, and I think we've discussed already a lot of compliance issues during this session. So the first thing that we need to understand while answering your question, I will say that Till date, the approach towards nitrosamines and NDSRIs has been more kind of a reactive approach as compared to being a proactive approach. That is, we have a product which has a nitrosamine or NDSRI exceeding the acceptable intake. It has to be recalled, and after recall, the reverse engineering starts. Rework or reformulation. So that is the situation predominantly that we have seen today. And the products impacted have been recalled, and we know what they are now. What's important is for companies and manufacturers to understand is in your product portfolio, what is the risk category of the products. The products have not been recalled. Perhaps nobody has done an extensive study of what kind of issues those products have. So it's important to have an understanding of the product portfolio, what is high risk, what is medium risk, what is low risk. Some kind of a risk categorization has to be done. Uh, what kind of NDSRIs? Well, I can tell you that in the coming years, we are going to see more surprises as more regions and more countries come under the scope of the nitrosamine regulation that I was earlier talking of. Uh, how to control this? See, I can give you two latest examples. There was earlier a communication from FDA with respect to presence of small nitrosamine in infusion bags containing certain drug products, and the manufacturers were asked to take cognizance of this and introduce mitigation strategies. Similarly, just few days back, there was another communication from FDA that this time is with respect to NDSRI, potential NDSRI, which is under investigation in drug device combination products of rifampicin. So if we try to, and the discussions that we are having, we know this, it's like opening up a Pandora's box and there's no end to it. But having said that, if you ask me from the compliance perspective, how things can be done differently, what I would suggest is that have a proactive approach instead of always being in a reactive approach. Design nitrosamine control strategies in your product development, product inception phase itself. Have experts on board, have cross-functional teams. One person cannot resolve this problem. You need to have a dedicated nitrosamine team within your firms, not for one submission, not for two submission, but throughout the product life cycle. So this submission and the strategies that we are talking is not once a time kind of an activity, but it needs to be monitored as long as the product is in the market. So have dedicated teams, have a nitrosamine project lead. There could be cross-functional members, not only from formulation, synthetic chemistry, analytical, but also toxicologists. There would be people who from third parties like external consultants or CROs or CDMOs that you can have expertise and you can utilize their expertise. So it's important to have a cross-functional team. It's also important to have understanding of what kind of changes you are doing to your process. So when we talk of a long-term perspective, the product shelf life, there's a concept of change management. It's very unlikely that nothing is going to be changed. You have a product for 10 years, 20 years in the market, and it's very unlikely that nothing is going to change. So change is inherent, it is inevitable. So but how do you manage the change? So change management would be very important. What are you changing? Why you are changing? What is the impact of change? These are very standard procedures, in manufacturing environments. So we need to adopt all these concepts when we are talking of nitrosamine control strategy. It can be a part of the broader contamination control strategy because what we need to understand is this is not just a technical issue or a regulatory imperative, but there is a patient at the end of the supply chain waiting for his or her dose of daily medication. We've seen most of the medications products impacted have been antihypertensive, metformins, and so forth. So these are products which are consumed by masses across the globe, almost on a daily basis, once a day, twice a day, depending upon the condition. So we need to understand that there is a patient at the end of the supply chain waiting for his or her dose. So we need to be proactive in executing this, and it is also important. We are seeing ICH guidelines, ICHQ8, ICHQ14, quality by design for products, quality by design for analytical methods. Even these concepts need to be included in the product development or method development aspects. So I think that is something which is important, and it's equally more important to be in a state of constant engagement and collaboration. You cannot be sitting in silos and say, hey, I will solve this problem. No. You need to engage with experts, you need to engage with pharmaceutical authorities, with the regulatory agencies, and be in a state of dialogue, because that is how you learn, and you learn to resolve the issues. Knowledge management, agencies talk of, knowledge management, and that is very important. There are so many updates or so many changes happening with respect to the technical aspects, the reg cmc aspects, so it's also equally important to have the right regulatory intelligence available at the right point of time. So these are some of the strategies that I feel can help. But if there's no answer to it, that what kind of products or what kind of NDSRIs would come in, as we work ahead and we try to learn and understand the formation pathways, the degradation pathways, I think there will be a lot of more learnings in the near future.
And just a very, very
Harry? Yeah,
sorry,
go
I
ahead.
just say that I completely agree with everything Arancha has just said there. I think probably the most important one is, as soon as you know what your candidate drug is, work out if it's a secondary amine, what its CPCA is,
Yes,
and do that early, because, so many
yes,
things I deal with are when people, organizations have, often tried in and to address issues, and it gets to very late on in development. And, if you're phase 2, phase 3, and you've got a category 1, category 2 nitrosamine, that's a little bit late to solve the problem.
Absolutely.
I
So,
completely agree. And also, when we are talking of CMC control strategies, of course, the mandate from the agencies with respect to confirmatory testing, which is an end product testing, but it would also be advisable to think about the critical process parameters earlier on and devise some kind of online or inline measurement strategies, so that you have at least some control over your reaction conditions. What's the pH? What's the temperature during the formation? So these kinds of approaches are also important. So when we talk of nitrosamines, from the GMP perspective, we say corrective and preventive actions, no? Kappa. So the corrective action is the product is recalled, either you rework or reformulate it. If it is successful, fine. If not, the product would be discontinued. But so it's more important to focus on the preventive actions, and that is where you need to engage earlier on and start looking into each of the unit operations, right, from product development up to commercial launch. Each unit operation in your supply chain needs to be closely monitored and evaluated, and definitely this will call for investment in man machine methods, and but that is inevitable, that is very much required.
Andy, I just want to remind you, um, if you get to a phase 2 or a phase 3 and suddenly discover you've got a nitrosamine, it happens,
yeah.
and then you don't know whether to remediate or throw the product away. Um, but I would come back to the comment I made earlier. Please don't throw the baby away with the bathwater.
Mm.
And this is a real problem for big pharma. It's walking a tightrope. We're all used to the idea of risk analysis, but risk analysis is based on actual visible risk. When we're looking at potential risks, then the tools that we use are as inaccurate as the potential or the definition of the word potential. And this is again a plea to the innovators. Um, we're going to, look. We all know half the drugs that we have on the market today would never have got approval under the modern regulations. And I'm just looking ahead to see good oncology products. Okay, S7, you can argue you don't need, but it worries me that we're going to lose potentially good drugs which might be able to be approved not with an assessment of 70 years, but with an assessment. We look at valsartan as a very good example. It's based on a 70 years, 1 in 8,000 risk. How many people start taking valsartan at the age of 10, right? I actually take valsartan. First of all, I take one that I know hasn't got any nitrosamines in it.
I
But
could probably guess
secondly,
what that is, but yeah.
yeah, I'm not going to tell you where it comes from. Um, but secondly, I didn't start taking it until the age of 70. So um, if somebody has a cancer and their predicted life expectancy because of a cancer or any other disease, or let it be type 2 diabetes and metformin, you take all the metformins off the market, it would never get on the market. If you look at the problems that metformin, there's more publications on metformin in drug product than there is for any other product. Extrapolate that back to developing metformin today, most companies will probably abandon it. And I think that is a warning. We should be very careful not to prohibit or prevent development of new pharmaceuticals because of a potential risk of a nitrosamine.
I now have a vision in my head of all of you with basement labs where you go and test your own prescriptions for nitrosamines. But just to move on to one final question, and this is for everybody. So we'll go through it one at a time. Is there a future when nitrosamines become routine quality control issue and it's managed by any competent analytical chemist, or because it is so complex and because it feels like it's constantly evolving as well, is it always going to require specialist help, specialist task forces, and more of a kind of crisis alert mindset? Archana, let's start with you.
Well, Ian, that's an ideal situation that each one of us would like to be in, that it should be a routine test. But I don't see it happening immediately in the near future because, as I said earlier, we are transitioning from building the foundation blocks into a more kind of a structured regulatory framework. We're trying to understand still the formation pathways. We're still trying to sort out issues with methods. So in the long term, yes, this may be possible, but I would put it in this way: to make it routine, first we need to understand what can we do in our development processes, in our manufacturing processes. As I said earlier, what can we do routine to make sure that this kind of an issue can be prevented and it becomes a routine test. What I would like to communicate here is that instead of putting products off the shelf, think of strategies where the risk can be mitigated and the product can be on the shelf and help the patients. That's the basic purpose of each one of us in the industry.
Yeah, what's your take on that one?
I do think there is a path toward this becoming more routine, but I think we are not there yet. For the well understood classical nitrosamine, you can already see that transition happening. They are increasingly handled within the standard QC with established methods and reference standards. But what remain a challenging is NDSRIs and the unknowns, the structural diversity, unpredictable formation pathways, and very low limit. Still, I mean, they demand deeper expertise. So today, it often does feel like a specialist or task force driven space. But looking ahead, I see a hybrid model emerging. Routine cases will move into QC, but there will be always a layer of complexity requiring specialized input.
Andrew.
Yeah, I think for me, we're at a critical pivot point with this. Do we extend this further to other nitroso species? Do we continue with the limits that we currently have? And I think as we try to portray in the discussions today, we can't necessarily solve the current issues purely from a CMC point of view. And actually, we don't even know what some of the risks and hazards are at the moment. We're still investigating some of those. So I don't see any way that this will be routine if we continue to assume, particularly for NDSRI's control, to levels, an order of magnitude below the simple TTC. If we're still going to talk about NDSRIs requiring control to say 26 nanograms per day or even 100 nanograms per day, we will never make this routine. So for me, I think the critical thing is, can we get a better handle on what the true risk of these nitrosamines are, both from the safety science around the nitrosamines, the NDSRIs, and dare I even say it, some balance and context compare in terms of how much nitrosamines and other nitroso species we get exposed to by the mere fact that we live on a planet that has nitrogen oxygen in the atmosphere, because we know we see these nitrosamines in many other things, our food. We actually metabolically produce some of the simple dialkyl nitrosamines. Yes, I mean, the argument may be they shouldn't be in pharmaceuticals, but we need to get the right context with some of this. But I think the main thing is probably the advancements from the preclinical side of things and the modeling. That's what gives me hope that we might be able to move towards a more static, controlled business as usual position.
And Malcolm.
Oh, this one's going to be easy. First of all, I forgot to thank you for the plug for the external consultants. I think that's very good, and I'm sure Andy agrees with me. But my answer is quite simple. Is this ever going to become routine? Not in my lifetime. And that is simply the length of lifetime that's left based on 70 years plus, and the fact that it's a stability issue as well. So it's not a case. If you could, if there was a possibility of saying at time zero I release, then there would be a chance of it becoming a routine procedure. With all the analytical complexities and the problems of the methodology and getting QC mindset to do these things is not so easy. But it's not a QC problem, it's a stability problem. And that is not going to shift into a routine strategy. And I don't think we're going to generate enough kinetic data while I'm still actively working in nitrosamines to enable this to become a simple QC sign off. Thank you. Good night.
Excellent. We've got time, probably just about the time for one or two questions from the audience. And although having said that, this first question I suspect could take up all the time and probably another hour, because it's about regulatory divergence. For products marketed in the US and Europe, FDA and EMA have not always been aligned on acceptable limits or compliance timelines for NDSRIs. How should manufacturers handle situations where those two big regulators are out of step? And is there real convergence coming down the pipeline? I think, Alshana, I'm going to start with you for this one.
That's an interesting one, and each one of us have been facing it. Yes, there are different acceptable limits for quite a few nitrosamines with different regulators of the globe. The first part of the question is how is there a convergence? Well, I would like to say there is no convergence or harmonization as of date to this effect. Let me be very honest about it. And how do these manufacturers handle these issues? Well, there are different ways in which these issues can be handled, and that's how we have been handling it. And I think we can do a separate detailed session on how to handle these kinds of regulatory issues. But at this point of time, I would like to say yes, these can be handled. It's like kind of having, it's more of a regulatory approach, a dual filing approach that we have been doing across EU and US, and it is possible. It's not easy, let me tell you, but there's no convergence or harmonization happening as of date with respect to these aspects because each agency has its own perspective.
So it's kind of basically there's no kind of holy grail on the horizon where they're perfectly converged.
Not immediately, but one thing is that ICH M7 and CPCA approaches are being adopted by regulators across the globe, and we see a amend, amendment to ICH M7 addendum underway, which has already been published last year with respect to nitrosamine. It's a concept paper which has been published with respect to conducting risk assessments and control of nitrosamine impurities. 3 years down the line, I think sometime by mid-June, we should be accepting the first draft.
So we had another question. I think this is an interesting one. It's actually a very similar question to the one which came up yesterday. It's about the difference between how nitrosamines can be handled by large, well-resourced organizations versus smaller and mid-sized manufacturers without potentially specialist infrastructure. So what's the realistic prioritized path to getting on top of this if you are a smaller organization? Maybe Andy, would you like to
Yeah.
pick up that one?
Yeah, sure. I mean, I think first of all, the most important thing is to understand the regulations, and certainly you could start by really focusing on the EMA position, Q&A document, and the FDA position. That gives you a reasonable feel for where the regulatory authority sits. I'm going to give you a comprehensive. I'll tell you what Health Canada thinks and the nuances of that, or any others. That's a good start point. The FPA position paper on the workflows, I think, is an excellent tool for actually explaining what the risk factors are and how you would then go through the process. But in terms of doing the analytical testing, doing the structural evaluation, certainly any kind of modeling or anything like that, yes, it's very difficult if you're a small operation. I'm hesitant to say this, but I think it is a valid point, and Malcolm kind of alluded to. I mean, there are experts who can possibly help some of these smaller organizations fill the gaps. And I'm working with a lot of companies that look to do that, and because it is really difficult to be on top of an issue where like this. I mean, it was my full-time job for 6 years or so, close to it, in AstraZeneca, and as a consultant, now it's pretty much the bulk of the work that I do now, and a lot of that is trying to keep up to date and pace with what's happening. And so, yeah, I'm not trying to sell myself or consultants, but the trouble is, and it is damn difficult for smaller organizations with a handful of people, maybe a CMC department of 1 or 2 and a safety department of 1, to be able to properly manage this. And that's the reality of this. That's the difficulty.
I can.
And I think.
I can go along with. I can go along with that because I was asked by a small innovative company to review their nitrosamine risk assessment for their new product, and I gave them a quotation, and then they sent me a risk assessment of 184 pages. Now, I'm hoping that you're all laughing because I wasn't, because I'd given them a quotation expecting to see somewhere between 10 and 20 pages. And they had covered every single eventuality, including, by the way, cleaning validation nitrosamine level, with all the discussions about why it wasn't relevant. Now, I told them, calm down, calm down. You don't need this. Throw away at least 50 of these pages without even reading it. To which they said, but we've already submitted it, which is the normal problem for consultants, is they come to you after the event rather than before. I'm sure, Andy, you've had these problems. But it's this is an example of complete and utter madness. The one practical thing I would say to a small company is, and maybe people will disagree, don't try and set up your own nitrosamine or nitrite testing. Not until you've got a critical mass where, because it's a difficult technique, it's an expensive technique with traps, this, that, and the other. Find yourself a reliable CRO that can do it for you and will work with you, rather than just charge you for every analysis, even when they know that some of them are a waste of time. Because the price and the startup time for these things, these methods are not methods which are designed to be easily transferred. Yes, USP's got methods, the FDA's published methods, but it's not your standard, okay, we don't have an analytical R&D department, but QC will handle it. I can assure you that they will handle it straight into the grave.
I just
That's about.
wanted.
Oh, sorry, mate. Go ahead.
Let me just add to the point which Malcolm has mentioned about the risk assessment, and I completely agree that at present there is a lot of inconsistencies in terms of risk assessment, the way manufacturers were performing. And that's the reason I think USP has realized this gap, and we are coming up with a risk assessment tool for drug substance, drug product, and excipient. It is going to be an online tool which anyone can use for quickly doing a risk assessment for their process and for the product. So at present, it is in a beta testing phase, and I think we will be getting it very soon launched at USP website.
Excellent. That's where we've got to call it a day, unfortunately. We've run slightly over, but I definitely didn't want to step in and stop that conversation because it was incredibly interesting. Thank you all very much. I think one thing I'd like to mention, I think all of you have touched on this, is that there is an awful lot of expertise out there, whether it is consultants, USP, the wider industry, and this is a problem that we all work together on. So very, very important point. So that's it for this session. We have another session starting in about half an hour or so, which is looking at quality control and reference standards in the GLP era. That's actually 1:30 our time, so actually in about 10 minutes. Thank you again to our panelists. Thank you very much to our sponsors, USP, without whom this would not happen, and we'll call this one a day. Thank you very much.
Thank you.